Hello, this is Mr. Arthur, and this is going to be a short video in the first half of gene expression, which is key area 3 of Unit 1, the Higher Human Biology course. So, gene expression is another way of saying protein synthesis. If the gene is turned on, then the gene is expressed. And if the gene is expressed, then the ribosome will make the protein which that gene codes for. So to recap National 5, the DNA is the code of life. It's the genetic information. The DNA molecule is called a code because it has 4 different bases: adenine, thymine, guanine, and cytosine. We already talked about DNA structure, and we talked about complementary base pairs, so that the 2 strands that are antiparallel are held together with complementary pairs of bases: adenine pairs with thymine and guanine pairs with cytosine. When it comes to gene expression, we're not talking about the 2 strands, we're going to talk about 1 strand. And we're going to talk about the order of the bases in this 1 strand. For example, a strand could be ATAT, or it could be GTAT. All life on the planet is different because the order of the bases in each gene can be different, and the overall quantity of how many bases can also change. So a section of DNA which codes for a protein is called a gene. A complementary copy of that gene has to be sent to the ribosome. The molecule that does this in the middle is mRNA. So mRNA carries a complementary copy of the genetic code from the nucleus to the ribosome, and the ribosome uses that code to create the correct protein. So, gene expression, we're going to break into 2 halves, and this video is going to cover mostly the molecules involved in it and the first stage, which is transcription. So it's called transcriptions followed by translation. Different ways of remembering this: you can think about a doctor writing a script, which you then take to a pharmacy, and the pharmacist has to translate the doctor's writing into the correct medicine they give you. Another way of thinking about it might be that somebody writes a script, and then the actor's job is to translate that into the correct interpretation. So transcription comes first. So you write the script and later it's translated. So that's first of all the order. Okay, we're going to talk about 2 different molecules: DNA, which we've learned about already, and now this similar molecule, RNA. You need to know that there's 3 structural differences between RNA and DNA. Any question asks you to compare, you must mention both. For example, you could say that DNA has a deoxyribose sugar. That's true, but it's not a comparison. If you said DNA has a deoxyribose sugar, but RNA has a ribose sugar, that's a comparison. In terms of bases, the DNA bases you know, RNA has 3 bases which are the same, but RNA does not have a thymine. Instead, thymine is replaced with uracil. So when you're making RNA, if the DNA gene starts with an A, the RNA is going to start with a U, because A would normally pair with T, but RNA doesn't have a T, so we're going to put a uracil in instead. This is a common thing that SQ will do in past papers. They will give you a DNA sequence and ask you about an RNA sequence, or they'll give you the RNA sequence and ask you about a DNA sequence. So don't be fooled. Any multiple choice, if it's about DNA, check there's no answers with a U in it, and if there are some, rule them out, and vice versa. Third difference, DNA, as you know, is double helix, it's 2 strands, double-stranded. RNA is only single-stranded. This image shows this nicely. So the DNA you're familiar with on the left, our new molecule is RNA on the right. They are both made up of nucleotides. The only difference is that DNA is made up with DNA nucleotides and RNA is made up with RNA nucleotides. And we can see an RNA nucleotide here on the right. So see, it's the exact same shape, still has a 3, still has a with a sugar, still has a 5 end with a phosphate. The only difference is that it's not a deoxyribose sugar, it's just a ribose sugar. Now, RNA itself, there's 3 different types of RNA. mRNA, you should know from National 5, and now at higher, we're introducing you to tRNA and rRNA. Doesn't matter if it's M, T, or R, all RNA is single-stranded, all RNA has RNA nucleotides with a ribose sugar, and all RNA has a uracil base instead of a thymine base. Okay, so the 3 types. The mRNA one is the one you're very familiar with. So the National 5 thing is still the same. Its function is to carry a copy, a complementary copy I should really say, of the DNA code from the nucleus to the ribosome. The second sentence is your new information at higher. Every 3 bases in RNA is called a codon. And that's really important because although the order of the bases codes the order of the amino acids, the way this works is we read that code in threes. So a triplet on the DNA, so for example, A, T, A, would then create an mRNA code, which would be, instead of ATA, would be UAU. That triplet on the mRNA would be called a codon. I'll skip to the bottom in the blue. We have a tRNA. The T stands for transfer. The job of tRNA is to transfer a specific amino acid to the ribosome. So the tRNA molecule you can see in the far right here, although it's folded up into almost like a T shape, it's still single-stranded if you look carefully. There's a 3' and a 5'. It's folded because of hydrogen bonds between complementary bases. And if you look closely, you can see the A is pairing with U. At the 3', it's what we call an amino acid attachment site. That's where the different amino acids that tRNA molecules carry will attach. And at the very bottom, although the rest of the bases are facing in the way and there have hydrogen bonds between the base pairs to create the fold, at the bottom there is 3 bases exposed. This is, instead of calling it a codon, we call it an anticodon. That's because the anticodon will match with the codon on the mRNA. So if an mRNA has AAA as a codon, the matching complementary anticodon will be UUU. That means that the mRNA and the tRNA molecules are able to join together using complementary base pairs. And this is really important when it comes to translation. The third type of RNA is the simplest. We don't need to know anything more about it other than the R stands for ribosomal. Therefore, RNA is part of the ribosome. So a ribosome is made up of 2 parts. It's made up of protein and it's made up of rRNA. And you just need to know that fact. Okay, so let's talk about the first steps. So we're going to look at transcription, the stages of transcription, the enzymes involved, and what we're going to call introns and ectrons, and how one of these are removed through a process called RNA splicing. So gene expression is the whole process of making a protein from the genetic code in the gene. First stage we're going to look at is called transcription, and that's when we make our mRNA, which carries the code. Okay, now the good news is this is very similar. Transcription is very similar to replication of DNA, so there's less to remember. However, it also means because they're similar, it's quite common for people to mix up DNA replication and transcription. So look out for the differences as well. So we'll talk through transcription of the gene, then we're going to look at some RNA splicing, and the next video will cover translation. Okay, so transcription is making the mRNA, which is the middleman which carries the code from the nucleus to the protein. And here we can see transcription. So, first of all, our enzyme in red is still polymerase, but because it's making RNA, it's not DNA polymerase, we have to call it RNA polymerase. And in the center there, step 1, the RNA polymerase is going to unwind and unzip the DNA molecule. So it unwinds it, it unzips it by breaking the hydrogen bonds, and that exposes both strands. Okay, so the bases are exposed. Now we're only going to deal with one strand, remember. There is no primers involved in this process whatsoever. Okay, so do not mention primer just because it's similar to DNA replication. Okay, it's similar because the RNA polymerase is going to work its way along the DNA strand from the 3' to the 5', and as it works its way along, it's adding and joining nucleotides. But this time, it's adding and joining complementary RNA nucleotides.
The primary transcript is created by RNA polymerase adding and joining together complementary RNA nucleotides.
So if the gene has the base sequence GCTA, the primary transcript will start CGAU. Once it's copied the gene and it reaches the end, the mRNA can detach. The DNA will then rezip, so the hydrogen bonds and the opposite strands will connect again and it will rewind back up. This initial mRNA molecule that's been made is what we call the primary transcript. It's called a transcript because it's made for the transcription, and it's called a primary transcript because it's the first version. This actually is edited before it leaves the nucleus. Okay, so you can pause here and read through what we just talked about, and then press play when you're ready. This diagram shows it in a sequential events, okay? Number one, you don't need to know about promoter regions, so just ignore that. The main point is that, start from number two, RNA polymerase unwinds and unzips the DNA. The RNA polymerase then adds and joins together three RNA nucleotides using complementary base pairing. That creates the primary transcript of RNA. That mRNA transcript becomes detached and the DNA joins back together, so it rezips, or the hydrogen bonds reform between complementary bases, and it winds back into double helix shape. So that's as done with transcription. Okay, so once the mature transcript's been formed, it has to go through this process called RNA splicing. That's because even though it's made a copy of the gene, there's little bits within a gene which are actually non-coding and have to be removed. For example, if you have like ACG and that's one amino acid, TTA and that's one amino acid, AGC, that's one amino acid, that's code. But then there might be a section that just says A. Now that's clearly not a code, it's just a repeat. That has to be removed. Okay, so RNA splicing's job is to remove non-coding regions. Now these non-coding regions have a name and the coding regions have a name as well. So we need to be clear which ones are removed. The easy to remember this is they're called exons and introns. Introns stay in the nucleus, exons exit the nucleus. So exons are the code because they exit the nucleus to the ribosome. Introns are non-code because they stay behind. Okay, there is two steps to describing RNA splicing and you must describe both. First of all, you see that enzymes remove the introns from the primary transcript. The second thing you have to say is the exons are then joined together. So the introns are removed and the exons are joined together. When they're joined together, this forms what we call the mature transcript. So this diagram shows at the start, you have the gene, a primary transcript is made, and a primary transcript contains both introns and exons. RNA splicing then takes place, so RNA splicing removes the introns, which are the light grey sections. These are removed because they're non-code, and they're called introns because they stay behind in the nucleus. And finally, the exons, which are the coding regions, they are joined together. That new molecule is called the mature transcript, and the mature transcript only consists of exons. Now, there's actually something called alternative RNA splicing. It's the same process with a slight twist. Although in Natural 5 you're taught that one gene equals one protein, this isn't actually true. And one gene is actually able to express several different proteins. You might think, how is that possible? And it's because of alternative RNA splicing. The second bullet point is the quotation you need to memorise and write if you're asked to describe alternative RNA splicing. Right, so I'll go describe it to you, then we'll go over the SQL language. So at the top, we have an mRNA transcript, okay? So it has introns and exons. It's still the same thing. The introns are removed and the exons are joined together. The only difference in alternative splicing is which exons are joined. So here you can see it goes red, yellow, green, blue. So we can remove the introns and then we can join together the red, the yellow and the blue, but we're not retaining the green. Therefore, we've actually just changed the genetic code. So therefore, that protein which is made at translation is going to be different from the other alternative mature transcript which has been made. Because in the right, this time we've retained the red, we've not retained the yellow, we've retained the green, and we've retained the blue. So therefore, our two mature transcripts, which have been made from the same primary, are different. And they'll both code for different proteins, because both mature transcripts now will have a different base order. So the two proteins will have a different amino acid order. Now, the tricky thing to word this is, if you imply that the order of the exons is changing, you'll lose the mark. So you'll notice in this diagram, we've not put the blue before the green or the green before the yellow. The order is the same. All we're changing is which ones are kept. So to answer this question, memorise that second bullet point. Start off with the end. Different mature transcripts are produced from the same primary transcript depending on which exons are retained. That describes RNA splicing. That's the answer to how one gene can code for more than one protein.